Thanks, Joe, and Julie and the whole team. It's great to be back here again. I was thinking how many times I've been to this conference, and I think I've been to all 5, I think.
Get you a jacket.
And it's really nice to be here and see many familiar faces again and talk about PCDH19 clustering epilepsy, which is the name I'll use today, even if it's not perfect. So what I thought I'd talk a little bit about is we've been working in this field for a while, so I want to sort of take you on a bit of a journey of our research and what we have done in the past, what we're doing at the moment, and a little bit about where we think we're heading to the future. Therapy has come up already in the context of this meeting, and I'll touch on that towards the end of the talk and talk about some different approaches that actually have been discussed already earlier as well. So I should say I'm from the University of Adelaide. I'm the other Australian in the room. We've heard an Australian accent already, and University of Adelaide in South Australia. Okay, so I'm going to cover a little bit of this territory, which we've covered already. I'm going to go through this fairly quickly because we're all on the same page, I think. So we're talking about
Just to check, I wanted the webinar seeing this in view. I just want to see how we can get on that view and say use slideshow. There we go. Sorry.
Okay. Great. Is there any way I can get rid of the control bar?
Brilliant.
Okay, thanks, Joe. Okay, so we're talking about a gene that lives on the X chromosome. There's 2 X chromosomes in females and 1 in males, of course. And this mutations in females, so in one of the X chromosomes, causes PCDH19 clustering epilepsy. The gene was identified by Joseph, who unfortunately can't be here, Joseph Gex, who's worked, as I said, in this field for a number of years. He had another competing meetings, and he's also from Adelaide, Australia. And he identified the gene back in 2008. So it's been quite a long time, although not as long as SCN1A, that we've known about this genetic form of epilepsy. I think the key thing that always stands out to me is the variability of the condition. In this case, there seems to be early onset seizures seem to be a feature that is common and can reliably be found in these individuals who are affected, and sometimes intellectual disability and other comorbidities such as autism spectrum disorder. So it boils down to really this heterozygosity of females. So that causes a mismatch in the brain, as we've been talking about. I'm going to give you some more examples of that as well. But importantly, also males with a mosaic deletion effectively causes the same thing. A mixture of cells in the brain that either have PCDH19 or don't have PCDH19. And in terms of the genetics, the way this plays out, I think, and someone can correct me if I'm wrong, but I think this is a unique form of inheritance. I haven't seen in the literature anything that's identical to this very unusual pattern of inheritance, where the males who carry the defective gene on the X chromosome are unaffected themselves, but can transmit it to the female offspring to cause the condition, and then these females can also transmit it to the next generation. And if they have daughters, there's 50% chance of them being affected and 50% chance of males being carriers. Very, from a genetic standpoint, this is very, very unusual. We've also seen this already, so I won't sort of spend a lot of time on it, but just to say that it's quite a large gene as genes go, and has this huge exon 1, which carries the load in terms of what's happening on the outside of the cell. And it's a number of repeats, these so-called extracellular repeats, which perform a function that is involved at the very least in causing cells to adhere to each other, to stick together. And there's also parts on the inside of the cell, which we probably know a little bit less about, but there's certainly an ever-increasing amount of literature on the function of this really interesting and important molecule. And as we've already seen as well, just briefly, many of the disease variants are located in this critical exon 1, the part that's on the outside of the cell, codes this part of the protein. And we think basically most of these are causing the kinds of changes in the protein which causes it to lose function completely, or at least partially. And I guess unlike many other disorders, there's no real standout common recurring variants for this particular form of epilepsy, and instead we see many, many different variants. So typically, it's difficult to predict exactly which variant is giving rise to someone in a family without screening specifically for a mutation in this gene. And in terms of function, as I said, there's ever-increasing amount of information on what this gene is doing. We've already heard about some of these already, some really nice work looking at its association with NCAD in the mossy fibre context. And also we've done a little bit of work on other partners that are likely also interacting with protocoderone 19 in the brain during development, other members of the same family, and I'll come back to that in a minute. So that's all happening on the outside of the cell. It seems to be important for cell migration and sorting by acting as a kind of a sticky molecule, an adhesion molecule. And on the inside of the cell, there's also a number of functions associated with this part of the domain, either through signaling or changing gene expression, some work from Joseph. So essentially all this put together is that we know this molecule is important in the brain during development and also after development as the cells and the neurons start to talk each other and form critical circuits. So when we started working in this area, we weren't working completely blind, but we didn't have very good tools to understand where the protein was expressed. I've just told you what the protein looks like. We could predict that based on the sequence of the gene, but we didn't know which cells had activated that gene during development. So that's a critical piece of information to understand how this particular condition is working. So a few years ago, we made a version of a mouse which, because we lacked antibodies to detect the protein, we made a version of a mouse which has a little tag on it, which enables us then to specifically use that tag to identify the cells that express protocoderone 19. And it really has quite a striking pattern of expression that we've seen earlier. This is very consistent. It's expressed in the cortex, in those layer 5 neurons, and also to some extent in layer 2-3 neurons, as well as in the hippocampus, in the postnatal brain. And this was consistent with another piece of data and another mouse model that we had, which this is a form of a mouse. It hasn't been described, actually, this particular mouse earlier today. This one has a different marker though, so it lacks protocoderone 19, but instead of expressing protocoderone 19, expresses this so-called LACZ gene instead, and this gives rise to a nice blue stain when we treat the brains with a particular chemical. And you can see it matches very nicely what we see with the normal protein. So we're starting to build up some information about where the gene is active, and clearly it's expressed in the brain as we would have hoped, in quite specific areas of the brain. And having these two different mouse models now meant that we could start to investigate what happens in the brain of heterozygous individuals. In other words, make a mouse that's got the same genetic makeup as affected females, females that are affected with protocoderone 19 epilepsy. And so we did that really simply by crossing these two mice together, and we've got two X chromosomes now, one with a wild type normal version of the protein that we can see with this tag, and the other one has this LACZ stain. And you can see very nicely, we see this quite striking segregation pattern in the developing brain. So this is before the mice are born, this is during development, and the cells essentially sort themselves out into either those that have protocoderone 19 or those that don't. And as we've heard many times already, this is a random process. So each brain is going to be unique in terms of the pattern, this sort of zebra stripe pattern that we see. Each one is different. And I think that really does play into the variability of the phenotype that we see in the girls who are affected. And for that matter, mosaic males as well, because they'll have a unique pattern of inactivation. Or a unique pattern of positive and negative cells, I should say. So here again is a little bit more detail on this. We can see so this part is going to be the cortex that we've heard about earlier, and this part is going to be the hippocampus. So when we looked at these individuals who are heterozygous, so in other words, the correlate of the human condition, and we let them develop into adult mice, and we looked at their brain patterns in terms of the broad amount of activity that was present within the brain, this EKG analysis. We found, interestingly, that we could see these spike wave discharge patterns that were elevated and more frequent and higher compared to either individuals that had only normal protocoderone 19, or interestingly, female individuals that completely lacked protocoherrin 19. So this again fits with this idea of the mixture of the cells being in the brain causing the phenotype. Now, what we don't know, of course, is what happens between this early stage of brain development and in these adults to cause this phenotype. And that's essentially the mystery that we want to get to the bottom of. And we've had some really nice discussion already and some nice data showing that's giving us now some real insights into this process whereby the brain behaves normally when it's got this mixture of cells in it. So we want to focus in, and the other thing I should say, like others have said earlier, we don't see seizures in these mice. We don't see frank seizures, and we have tried to induce seizures using the same kinds of techniques that we've heard about earlier, and we're going back to a thermal seizure assay as well to see if we can get that to work. But it's really a case where the mice, and as is commonly found, mice don't tend to develop seizures as readily as we see in humans, generally speaking. Okay, so we got interested in this. What's happening here at the border? Why is it that the cells that express protocoherrin 19 here like to stick together, and those that don't express protocoherrin 19 like to stick together and effectively segregate away from each other in this unusual, abnormal way during development? Why is this occurring? So we started to look at a little bit more detail at the molecular level to see what's going on. So we knew from previous work that protocoherrin 19 is expressed in synapses. So we decided to have a closer look at that part of cellular interaction. So once the stem cells in the brain form the neurons then start to talk to each other and form these synapses, as we've heard earlier on today. So we wanted to know whether protocoherrin 19 was at synapses, and if so, how often. So what you're looking at here is neurons that are grown in culture. This is the cell body, and these are the processes that come out of the neurons. And we have taken these neurons from our tagged brain, so we can use this staining technique to see where the protocoherrin 19 is in the neurons. And we've co-stained them with another neuronal marker, SYN1. And you can see from this graph over here on the right side, and also from these co-localization studies, that there's many examples of where protocoherrin 19 is present within the neurons. So then we wanted to really start to sort of tease apart what's happening at the neuronal level to determine whether, if we have a protocoherrin 19 positive neuron and a negative neuron in the same dish, would they talk to each other normally, or would they find themselves less likely to talk to each other via these synaptic contacts? So in this experiment here, we've got, if you like, a carpet of neurons shown here in red. They're wild type neurons, so they've got intact protocoherrin 19. And into them, we've engineered some knockout neurons. So these are the ones that don't express protocoherrin 19 using a technique that effectively disrupts the protocoherrin 19 gene. And we've labeled these with green, a green fluorescent protein. And then essentially what we do is we do the same experiment. We label the cells in green, but we leave the protocoherrin 19 intact as a control. And that's what's shown here on the left-hand side. This is the control, and these are the cells that have the mismatch. So if you like, this is like the disease correlate, and you can see we've got significantly less contact between the cells that at the neuronal level, at the synaptic level, between the cells that express protocoherrin 19 and those that don't. To really try and nail this down a little bit further, we performed an identical experiment, but in reverse. So now we've got a carpet of neurons that are lacking protocoherrin 19. And then we've introduced the protocoherrin 19 gene in those rare cells that sit on top of them. So again, we see this is the mismatch condition, and we see exactly the same phenotype. Again, underlining the fact that cells that either, when we have a mixture of cells with and without protocoherrin 19, they are less likely to talk to each other via synapses, or at least make contacts via synapses. We wanted to then look at that at a more sort of broader level to try and get an idea of what this really means in terms of network activity. And you can do that again using cells in culture. So these are neurons grown in culture. We've got either wild type cells, we've got knockout neurons, or we've got this mixture of wild type and knockout neurons in the same dish, which is effectively the disease correlate here. And when we look at the network activity, so in other words, broad network activity, how these cells are talking to each other across the dish, you can do that using this so-called multi-electrode assay, or MEA. which effectively has these electrodes scattered across the dish, and it can measure the amount of electrical conductivity between these cells, measuring things such as burst duration, number of bursts, the time between bursts, etc., etc. And really interestingly, when we do this kind of analysis, this is the mismatch condition shown here in red. You can see there's a number of features of this network activity that are different, significantly different compared to the homogeneous cultures of either wild type or knockout neurons. Again, building evidence that it's the mixture of cells that's causing abnormal network activity. To go back in vivo and look at this a little bit more carefully, we have performed an additional experiment. And the idea of this experiment was to again introduce a condition where we're looking at positive and negative cells, but this time in vivo. So with this experiment, what we've done is we've labeled cells, the cells that are going to express protocoadherin 19 by making them express a green fluorescent protein at a particular, by introducing that condition during the development of the cortex. We know that the neurons are born at around 13.5 days of development that express protocoadherin 19. They're the ones in this layer here, as we've seen already. So we can engineer them to express green fluorescent protein using this so-called in utero electroporation approach. And you can see the green cells here. So the idea of this experiment was to make the protocoadherin 19 cells express green fluorescent protein, and then ask what happens to the processes of those cells, and in particular, can they cross over to the other side of the brain, and will they make as many contacts and cross over to the other side of the brain as frequently when they're encountering other neurons within the developing brain that don't express protocoadherin 19. And again, that's shown here. This is the data. This is the control condition here, and this is where we've introduced the mismatch. And again, you can see that by introducing cells that don't express protocoadherin 19 into this system, we again see less activity, less connections between wild type and knockout cells. So there's now a lot of evidence, and of course many other studies as well, that have examined this property of mismatch between wild type and knockout protocoadherin 19, and the fact that it compromises interactions between cells at the synapse level or at the interaction level. We wanted to probe the details of this in a little bit more depth, and to do that, we have looked more broadly at this family of molecules that protocoadherin 19 belong to. So I need to step back for a moment and tell you that protocoadherin 19 is just one member of a group of proteins that all have quite similar structures, but they're subtly different, and importantly, they function slightly differently. And these are called the so-called non-clustered protocoadherin family, of which there are many members. And here's just one, protocoadherin 19. As you can see, there are many members of this same family. Very interesting and important family, and very complex in their expression and function. So these have been studied for a number of years, and you can see here, looking at this expression analysis, this is essentially taking embryos and looking one by one to determine where each of these family members are active during development. You can see they're all expressed in the developing brain, and if you look carefully, and if you really drill down on which cells are expressing, it's a very complex picture that emerges. A lot of overlapping expression and probably a lot of unique combinations of expression that we think are probably important for the way that the neurons are wired during and after embryonic development. And this has also been examined to some degree at the molecular level by looking at a related family of cadherin proteins, the clustered protocoadherins, which we don't have time to talk about today, but some really nice work that came out a number of years ago. Came up with a model which essentially said that combinations of these proteins expressed at neighbouring cells act in concert to cause either interaction, or if there's a mismatch in this combination, lack of interaction between the cells, which fits perfectly with the kind of, with our thinking around protocoadherin 19. So we wanted to probe this a little bit further, thinking about now if we can put protocoadherin 19 in the context of these other factors to ask the question, what happens in combination with other members of the same family, and can that inform us about the way this protocoadherin 19 epilepsy is caused in terms of brain development? So this is a pretty complex slide, and essentially what I'm showing you here is about 100 genes simultaneously overlaid in their expression in the brain. So we did something called PCI-seq, which is a fancy way of simultaneously looking at gene expression. So just to go back, this is a kind of one-by-one analysis. Now what you can do with this more modern technique is to use the same brain section and look at many genes simultaneously in the same brain. This approach is probably a little bit more sensitive than RNA-seq, which is another sort of similar approach, and it preserves the spatial location of the genes within the brain as well. So it's a sort of a complementary analysis to RNA-seq, which will be familiar to some of you in the audience. So what we did is we looked at all the members of the protocadherin family and a number of other markers as well, which we felt were important for the analysis, for example, excitatory neuronal markers, and drilled down a little bit more carefully on where there was co-expression between protocadherin 19 and these other markers. And really sort of to cut to the chase, we found that protocadherin 19 interacts with it in the cortex often. So when I say interacts, I should say overlaps in its expression with protocadherin 9. And in the hippocampus, we found also protocadherin 9 and 1 were overlapping expression with protocadherin 19 as one of the major sites of overlapping expression. We hadn't thought about these molecules before, and we decided, therefore, to look a little bit more carefully at what might be going on in terms of this functional interaction, whether we could find evidence that, in fact, these two molecules were functionally interacting. So we did this using co-IP, and this was also a technique that was mentioned earlier. And you can see by the fact that we've got bands in these tracks here that we can see evidence that these two molecules are physically interacting in cells. And the other molecule that we kind of drilled down with as a potential partner for protocadherin 19 is CDH. This is a cadherin 13. And the reason we looked at this particular one was that this gene had already been implicated in neurodevelopmental disorders. So we thought that was important to look at. And again, it interacts by co-IP. So that's all very well, just because you've got overlapping expression, and even IP analysis doesn't tell you very much about what is going on in terms of the function of the proteins. And one way to look at that is to use these so-called adhesion assays, which are essentially a way of determining whether cells will associate together if they express protocadherin 19s of the same identity, or whether they segregate away from each other if there's no interaction between them. So what I should say is, so this is how the assay works. It's probably just easier to show you on the assay. Typically, these have two different colors of cells labeled with two different molecules. This is just a control here to show you that when you mix them together and you allow them to either mix or spontaneously sort, if the molecule is the same in each of the cells of the different colors, the red and the green, then they'll mix nicely here, as shown in all of these cases. But when we've got different molecules within the different colors, then you can see we get sorting, which is this situation here, which means that the cells don't like to sort, to associate with each other. So you could use this kind of assay as a surrogate for what might be happening in vivo to test whether there's functional interaction between candidate molecules. And this became actually quite a complicated analysis in the end, because it wasn't a simple interpretation. But what we found, and this builds on similar assays that we'd published earlier with different members of the protocadherin family, but essentially what we found was that the cells didn't really care very much in this context whether they expressed protocadherin 19 or not, and that's shown over here. So in other words, we've got cells here with 19 and 1 versus cells with 1. They're pretty much associating in the same way as the control. But in the other two cases, we saw that protocadherin 19, when it was missing from one of the populations, as is shown here and here, the cells really didn't like to talk to each other. So this is providing evidence that a combination of proteins, either involving 19 and 13 or 19 and 9, creates a combinatorial code or an association that it's very important that there's the same combination on the other population, because if you're missing predicate hearing 19, you won't get the interaction as you should. So this has allowed us to develop essentially a working model based on, I guess, all of the data that I've been telling you about so far. And as I said, it's a working model. It's something we need to test in vivo to a greater extent, but it gives us some, I guess, some ideas, some plans to go forward with further in vivo analysis. So what I haven't told you, that I should have told you, was that all of the interactions, all of these molecules were also expressed in excitatory synapses as part of the original expression screening. So we think perhaps it's not just a matter of having predicate hearing 19, and we heard NCAD is an important partner. These are some other candidate partners that might also be important in vivo. Predicate hearing 9 and predicate hearing 13, in combination with 19, we think would help neurons to associate. If we lack predicate hearing 19, so this is a correlate of the hemizygous males, we can see that there's still molecules here that allow association to occur. But in the disease state, where we've got some predicate hearing 19 positive neurons and some that lack predicate hearing 19, we shouldn't get functional association between these, which will give rise to the condition. So in order to, as I said, in order to get further evidence for these kinds of models, we need to develop for these working models, we need to develop better mouse models to test these theories out. So this is quite interesting because this is exactly the same type of model that we've already heard about earlier. And we've engineered it, I think, completely independently. So this is quite interesting. It's great that it's working for you. It's working for us as well. So this must be a good idea. So just to take you through it, just as was mentioned earlier, this has a special sequence either side of the normal open reading frame in exon 1, such that in the presence of this Cre recombinase protein, it will remove this entire important part of the molecule, and it will now express this green fluorescent protein. So this is a way of enabling us to delete predicate hearing 19 with quite a lot of precision during mouse brain development. This is just, we haven't published this one yet, but this is just to show you that the conditional model, in other words, when we have it in this state, behaves like a completely normal allele in terms of gene expression and protein expression. Okay, so we've been playing with this system, I guess, to try and get a better idea of how well it behaves in vivo, and also to try and test some of our ideas around creating mismatches in particular populations in vivo and asking the question, is there a phenotype associated with that? So I've just got a little bit of data to show you, which is really work in progress around some of the phenotypes that we're seeing when we use different so-called Cre driver lines associated with this new flox-to-veal. So SYN1 Cre is one that's quite commonly used. It's expressed in the neurons as they develop from about 13.5 days onwards, as I recall. And when we use SYN1 Cre with this conditional allele, as expected, really, we get back this normal expression of predicate hearing 19 in the cortex, these layer 5 neurons, as we would have predicted. So this is quite an interesting model to look at, because it enables us to, as I said, remove predicate hearing 19 from a range of cells in vivo, but it leaves other neurons within the brain retaining predicate hearing 19 function. So you can think of it as a kind of extreme example of what we see in heterozygous females. So as I said, we're still exploring what this really means in terms of the phenotype, but we've been looking at other parts of the brain and asking, are they developing completely normally in the context of this deletion of predicate hearing 19 in some of the neurons. So we do see that hippocampal neurogenesis appears to be actually elevated in these mice, and that's shown here on the right-hand side. So these are two markers of hippocampal, in this case, proliferation. So this is Ki67. So we're looking at proliferation markers within the hippocampus, the learning and memory center, and a second marker, double cord, and as you can see, is also elevated when we look carefully in these mice. So this is Again, we don't really know the reason for this, but it's evidence that, again, mismatch within the developing brain is causing a phenotype. And then we've been looking, in this case, at a so-called open field test, where we take mice and essentially put them in a box, and we track their activity using a camera, and then we can then do analysis of those track paths to ask questions about how they're behaving in this environment. So again, comparing the control mice with these floxed Syn-Cree mice, you can see we are seeing in some cases, in this case, evidence of altered behaviour in this environment. But again, we haven't been able to really tie this back to the brain phenotype that we're seeing. We've also been looking at the hippocampus, and this time using a different driver line. This is so-called GFAP-Cree, where we remove protocoherin 19, this time in the hippocampus, essentially the stem cells within the hippocampus. And this labels a lot of the protocoherin 19 cells because of the expression. So the neurons therefore form within the hippocampus lacking protocoherin 19. So I think this would be a really interesting one to chat with about later, to see if we can, I guess, unify some theories around what's going on in these mice. But again, we see when we look at the hippocampus that they're not behaving normally. So double cortin again is elevated in these mice, and as Ki67. So again, evidence that if you create a mismatch between the neurons within the brain that either have or don't have protocoherin 19, you start to see phenotypes to do with neuronal behaviour and in the previous case, mouse behaviour as well. And then again, we're using behavioural assays to test this one. And again, it's pretty early days, but we are seeing evidence of significant differences in this novel object recognition test. So you can kind of think of these examples as extreme examples where we've really deliberately created two classes of neurons within the brain, some that have protocoherin 19 and some that don't. These differ really from what we see in heterozygous females in the sense that they're not random. They're very targeted, and I think because of that, there's a little less variability because we're not dealing with a random exon activation process. We're dealing with mice that have essentially the same targeting of the neurons in each individual. So there's probably a little bit more reproducibility with these kinds of experiments. This is just, I guess, a little bit just to show you that we've transmitted, I don't know if you've done the same with yours, we've just transmitted this mutation through the germline now, so we now have a knockout GFP line as well, which we tend to use rather than the laxed line. All right, so for this part of the talk, I think the conclusion is that this mosaic expression of protocoherin 19 is because it involves a random exon activation process in females, and it's also essentially random in somatic males, causes disruption of neurite synaptic establishment or communication between the positive and the null neurons, leading to abnormal network activity. And we're trying to sort of delve into the molecular details of that. For the rest of the talk, I want to focus in on, I think, two really key questions where we want to sort of put our efforts into the future. And that's to ask the question, at what developmental stage is the primary pathological lesion manifest? In other words, when is it that protocoherin 19 is really important for brain development? And this might be a large period of time, it might be a small period of time, it might be in many processes, it might be in a single process. We don't really know at this point. But of course, we've got more and more information coming out about the processes in which it is important. And really importantly, can we reverse the neurological phenotypes, perhaps by using a genetic approach to intervene and create, for example, completely null brains? So this now fits into, I guess, more broadly with what my lab does in terms of CRISPR therapy. So we're interested in using CRISPR-Cas9, a pair of molecular scissors, to see if we can come up with new ways of treating eye disease and muscle disease. And I'd always considered our protocoherin 19 epilepsy research kind of separate from those, but now we're starting to try and bring them under the same banner and think about ways that we could use CRISPR-Cas9 or antisense oligonucleotides. At least consider the prospect. Is it possible? Should we, is it a feasible approach? Okay, so why would we want to do this apart from the fact of the obvious need around the clinical side? The drugs, I gather from my reading, I'm not a clinician, but I gather generally or can have quite poor efficacy. And in any case, controlling seizure activity doesn't necessarily control the other behavioural cognitive issues that can occur with protocoherent epilepsy disorder. So what's really interesting about protocoherent 19 is that, as we've heard about many times, individuals that lack protocoherent 19, so in the human world, that's hemizygous males. As far as I'm aware, we're not, no one's ever seen a homozygous female human. I don't believe, but in any case, the important thing is that individuals that lack protocoherent 19, in this case, it's a mouse that's female, so with two X chromosomes, but both of them lack protocoherent 19, they have a pretty normal EKG phenotype, and by any measure that we put them through, they don't appear to have any compromised brain function. And because of that, we could consider perhaps an approach that was discussed briefly after the first talk today. So instead of adding N-cadherin, which was the elegant approach that we heard about, an alternative is to convert an individual that's heterozygous, so has the disease genotype here, into one that lacks protocoherent 19 completely, because protocoherent 19 is not essential for brain development, and individuals that lack the gene appear to have no phenotype. I'd really like to get some feedback on that hemizygous male lack of phenotype, because that's everything that I read tells me that, but perhaps, and there was a hint that maybe that wasn't completely the case in all, but I'd love to talk about that afterwards with anyone who's interested. So, can we control or alleviate neurological symptoms in protocoherent 19 by eliminating functional copy of protocoherent 19 in heterozygous females? That's the question. And the other thing that makes this, from a genetic molecular point of view, reasonably attractive is that, as we've seen already, there are many, many mutations that cause, or disease variants that cause protocoherent 19. But in this case, unusually, and so for many gene therapy projects, you're looking to restore the function of many different disease variants. Therefore, it becomes a very big and complicated approach. Here, though, we're looking to target the wild type allele, the normal allele, if you will, which everyone carries anyway, which there'll be a little bit of variation in, but not nowhere near as much as we're talking down here. So in other words, it could be a one-size-fits-all type approach. We've decided to look at, I think, a key issue, which is when does the pathological lesion first occur? And this is a really complicated one, and I wish I had more data to show you, but I'll give you, I guess, an idea of where we're heading with it. So as you've seen already, we have this nice so-called floxed allele, conditional allele in mice, where we can, with a fair bit of precision and timing, remove protocoherent 19 from the cell. And we can now, instead of using the types of approaches I described earlier, where we would take it out of a subset of neurons expressed at a particular time, we can have a more generic approach where, by using a system that's inducible with a drug, we can switch it off at different times during embryonic development. So this is a kind of different approach. Now we're talking about timing, and we're switching it off in all of the cells. That's just some controls. So this is basically the philosophy behind these experiments. If we start off with a healthy individual, we can, this is the genotype here, we can turn it into an individual that is then susceptible to protocoherent 19 epilepsy by adding the drug and removing it from some of the cells during development. So that's going to give us some idea about when the timing of the lesion and when it's important. And by the same token, we can do the reverse. We can start with a disease phenotype and then remove the protocoherent 19 from the floxed allele at any time during development and ask the question, can we rescue the phenotype using this approach? So this is basically what the lab is investing some time in. And what makes these so difficult is that we need to essentially do a time course across many stages of development to get a handle on when it's important to take away protocoherent 19, which is essentially what I'm showing you here. The other difficulty with these experiments is that, as we've heard about a lot already, this disease phenotype in mice can be variable and it can be quite subtle and difficult to detect. So again, another discussion point that I'd like to flesh out some more is, and we've already had some discussion around this, is what are good assays that we can use to determine whether the mouse that we potentially rescued, in other words, what are really good disease markers that we can test for in behavioural, probably behavioural markers for protocolyrin 19 epilepsy, to ask the question, if we've got them secure then and consistent, that means that we can then test with more, we can be more sure that any rescue that we're doing is having a significant effect. All right, so this is really saying the same thing. These experiments are quite difficult to set up because we need now a triple transgenic mouse, so these are quite expensive and time consuming, and you don't get many of these out at the end of the cross, and just really underlining the fact that this is going to be quite a large undertaking, but we have started. And this is to show you that we've got the system working pretty well now. So at least in terms of knocking the gene out during embryogenesis, this is just to show you that it controls. We start off with no, these are completely flocked. So in other words, we have intact protocolyrin 19, then we add the drug, and you can see very nicely that we get this pretty well ubiquitous recombination, which allows us to knock out the protocolyrin 19 and the green cells appear as we would hope. So the system's working. Now it's just a matter of going through the time course. And by the same token, that's going to tell us when it's important to have heterozygosity for protocolyrin 19 in terms of the phenotype. And by the same token, the reverse is just as important. Can we rescue this disease phenotype to a healthy phenotype by intervening at a particular stage of embryogenesis or postnatally? So this is, I feel, a very important baseline that we need to understand before we could even consider therapeutic intervention. So I'm just about finished, and this is just to tell you that we're also playing a little bit with delivery methods, because one of the difficulties is to deliver any therapy to a developing brain, be that postnatally or before. So we're talking about some really complex and difficult approaches here. So this is things that we're at least working up in animals, delivering it to the embryos, delivering CRISPR to the embryos via so-called retroorbital injection, and also to adult mice or postnatal mice, again, using retroorbital injection. So this part is to, you know, if we feel that we can rescue the phenotype by intervening at a certain time or before a certain time, then we also need to be able to deliver the tool to rescue that phenotype at a genetic level. So that's what we're doing here. All right, so this is the last slide, and I'll try and wrap things up. So protocolyrin 19 epilepsy, as we've heard, is caused by these unique patterns of cellular mosaicism in heterozygous females and mosaic males, and I really do believe that that is probably the root cause of at least most of the phenotypic variability that we see. What is the pathological cause? That's something that is complex, and we're having a great day hearing about some of the experimental approaches and advances around that. And when does it manifest? Because this is critical if we want to reverse it. Is it presynaptic? So in other words, is it really important that the cells segregate from each other in the developing brain before they form neurons, as we've seen with this kind of striped phenotype? Is that really important? We don't know, actually. Is the problem with protocolyrin 19 expression being mosaic at the level of synaptogenesis? It could be. We've got some evidence for that. Or is it to do with synaptic function, as we've already heard about today, and some really nice data? Or possibly it's a combination of the above. So we've got a lot of sort of functional studies to get through to try and get to the bottom of when and where protocolyrin 19 is critical. And if we can understand that, is there any prospects of intervening and rescuing the phenotype through in vivo deletion of protocolyrin 19 or other approaches that we've heard about? So we need to, I think, look at the models first and then ask that question before we can really consider taking that further. Okay, so with that, that's where the work's being done at SEMRI in Adelaide, and I would really, really like to point out two people who have been instrumental to this. work, really all of the work's been done by Stefka, who's a postdoc in the group and worked in this space for a number of years, with fantastic assistance from Mick, who's shown there in the top, Michaela, collaborators, Joseph, and others that have been sort of instrumental in our thinking around this field and have been really fun to work with over a number of years. And lastly, and certainly not least, funding from Predicate Hearing Alliance really has supported the studies, all of the studies that I've been telling you about today, and really got us going in a bigger way and enabled us to get larger funding that we wouldn't have been able to get otherwise without that initial support. So with that, I'll close and I'll be happy to take questions. Thanks.
So this is really exciting work. I mean, seeing the course of all this. I'm just curious in terms of timing and how medical trials, proceed, et cetera, when there might be something that fundamentally would affect any of the folks in this room.
Yeah. I would take a step back from that question because I'm not sure yet whether we know if it's going to be possible to intervene in a genetic way to rescue the phenotype. I think there are lots of really interesting things being tried. Dravet syndrome came up earlier. There's been some really amazing antisense oligo approaches in that space, but it's a different condition and it's caused a different mechanism. So I think for me, the most critical thing is, at least in an animal model such as we've been using, can we find evidence that we can intervene and rescue the phenotype? And if so, how early do we need to do that? Because PCDH19 is expressed very early on during development. It comes on before the brain is even formed, it's really just a bunch of stem cells. And it doesn't mean that it's required at that time or that the mosaicism at that very early time is important. It might be, but it might not be. So I think we just need to do the experiment to get some evidence about when we need to intervene to see if we can rescue the phenotype.
I guess the struggle there is the phenotype, right? I mean, because one could argue that if an ASO approach is safe and at least demonstrates a proof of principle on the mouse, that could there be exploratory human trials at some point. Yeah,
I agree. One thing that we have, all of us have faced as looking at models of PCDH19 is first of all the variability, because each heterozygous individual is unique. So we need to have large numbers to see an effect. And secondly, the mice particularly are not, don't have a dramatic phenotype. They don't see spontaneously. ECOG can give some insights, but you probably need large numbers. So I think all of us are searching for good assays, and there's been some described already: behavior assays, network assays, electrophysiological assays, anything that can give us a reliable handle on PCDH19 mosaicism and whether it's been rescued or not.
Really great talk. In terms of getting a phenotypic behavior assay, especially for your time course where you're rescuing different types of direct development. Have you or has anyone done any neonatal behavioral assays? I've performed those just to sort of get a quick and dirty answer into, is this mutation causing disease, particularly like ultrasonic vocalizations for early communication deficits?
Short answer is no. No, we haven't. We've only looked really at adult behavioural. We've collaborated with ECOG. We're doing some seizure testing now. Thermal seizure. We did some years ago and we couldn't find it working reliable. We tried PTZ. I think all the things that you've heard about, we've probably all tried similar you know, a similar range of phenotypes. I really thought that was great that you put a cohort of mice through 9 behavioural assays. That, I'm sorry, didn't see anything that was sort of stood out as a really reliable behavioural test. Yeah, I can happily chat with you afterwards about some neonatal stuff. That'd be great. Yeah.
Obviously, sorry. Obviously, what you're doing is amazing and sounds like it'll be hugely beneficial for the future. Not our babes right now. So admittedly, I feel a little bit deflated. I was hoping that you'd come with something a little more sparkly for the here and now. Without wanting to insult you, but not having anyone else to ask, how do we know that you guys are the front runners in regards to research? How do we know that there isn't another research body that's maybe going down a different line of inquiry and is perhaps ahead of you with an ASO?
It's all right, I'm not insulted. No, it's a good question. And look, I felt it was very important to be, I guess, completely honest in my assessment, in that I don't want to say anything that would be, you know, put too much of a shine on the situation. I'm just trying to, I guess, lay it out as I see it. The possibility that there are others out there with more advanced. One thing about science is you never know. You know, you just never know. Any day you could get a paper come out from a group that you just weren't expecting. So, and possibly companies as well. So, it's possible. I think, you know, in this room you've probably got some, you know, probably I would say the most prominent people in the field that I know of. That doesn't mean there aren't others out there working on this as well. And of course, the way science works is that you don't hear about what they're doing until it's basically the end of the story and it's published. So I too hope that there's going to be some really exciting developments happening that I don't know about because of the need. But by the same token, I'm not aware of any that probably aren't in this room, I would say.
Yeah, maybe just to add to that, I mean, Julie and I have heard of Praxis that had an ASO a couple of years ago. It was in a slide deck, but I don't know what happened. So often, you know, when things look good up front, then some real work, I won't say real work, but it gets into the hands of people who know how to present or how to look at proof of principle and it falls flat. It doesn't mean it's not something there. But then as a clinician, right, then we have to take the step of just because something looks good in a mouse, then taking the leap, right? We tried to do this clinical trial 5-6 years ago, ganaxolone, and just the, you know, the population in terms of meeting a regulatory approval here in the US and the FDA is another hurdle, right? So there are many hurdles, and I'm not trying to say yeah, but there are. I feel like it's moving. The fact that there is a gene and that there are people that are looking at it, and now that this ASO platform is expanding in other monogenic epilepsies, that yes, this isn't going to happen in the next 3 years for PCDH19, but things are moving faster than I thought they would, even 5 years ago. It's never fast enough. I completely am sympathetic to that. Julie.
I don't want to burst your bubble completely, but I mean, I think several of us are looking at ASOs, and they're unfortunately not as simple as we'd like them to be. So I didn't have any of that data to present for you today, but we have not given up on this approach. It's still in the works in both human tissue and mouse as well. So I'm hopeful maybe the next meeting we'll have more ASO data to show you all. But I'm really happy that Dr. Thomas's group is doing what they're doing right now, looking at timing and how important timing might be, because I think that's going to be one of our big questions is when is it appropriate to intervene and can we actually pursue the phenotype? Because as we talked about, PCDH19 probably has many more functions than just making the striking pattern. So if we rescue some of those functions later on, Is that going to change anything, or do we really need to rescue very early?
I'm wondering if all three of our, we talk a lot today about the adhesion and the striping, and then there's those, 25% of patients that do go into remission, and some hypotheses surrounding male shift pattern and steroidogenesis. Is there any way that those two theories come together in terms of why adolescents are some? Presumably these protocadherins are still being expressed in the same way, and the striping is not changing in a teenager.
I don't have an all unifying theory for you, Joe. And I think one of the reasons that we discussed a lot with Joseph, who's been working a lot in that area, one of the difficulties is that the genes that are in humans, in mice versus mice for the steroidogenesis pathway are quite different. So it's very hard to correlate between the two.
I think all I can say about that is that the stripes are probably not the whole answer, right? So there's probably some neuronal signaling pathways that are involved as well that may be more modifiable in later years that may be applicable. But it's a really complicated gene, as I think we've all expressed today, and that I hope we're all up for the challenge of trying to figure it out.
But basically, it's like a phenotypes are like based on synaptic functions, and synapse formation is post-natal, meaning afterwards. So that's why, even if like a cell aggregation abnormality happened during development earlier before birth, if we can correct synaptic connections, synaptic patterns, the hope is that we can treat patients, meaning after birth, and that's what I believe can happen.
And I just want to add, as you're developing assays to figure out as markers for whether you've rescued it or not, it is more than just epilepsy, it's behavior, it's sleep, it's a whole lot of things. And as a parent, maybe you can't rescue the epilepsy portion of it, but if you can rescue the behavior and learning, it's still worth doing. And so if that's not the marker you have, then maybe it's difficult, but I just want to keep in mind that it's not. Or if you can't do the behavior and learning, but you can stop the seizure, that's huge too.
Mm-hmm. Yeah, no, I got to take that on board 100%. I would be happy if we had a reliable marker that spanned any of the phenotype, but we're not just looking at the seizures either. So we'd be very happy if we had a good behavioural test that we could then turn to say, okay, we can rescue that. That would be great.
And then maybe one final question I had about ASOs and just the genetic. We have one?
Oh,
sorry. Excuse me.
As we're looking at the changes, especially in adolescence, and the migration and regeneration and all that, is it possible that the synaptic pruning is impacting some of these cells and what gets put where and where we're losing some of the stuff for our kiddos?
It's possible, yeah. So there's really, and because PCDH19 spans all of these processes, it spans the developing. So the cells are neural stem cells to start with, and they turn into neurons in this layers in the cortex and also hippocampus. It's expressed then. It's also expressed in some of the cells as they migrate to their final destination, and it's also expressed in the synapses that occur between some cells. So we really just we don't know what the critical processes are and when they occur. And I hope I agree with Charlie that if it's down to synaptogenesis late function, then that's going to be the best case scenario. And I really hope that actually that the stripiness in the brain isn't the whole story. In fact, isn't that important at all. It may be a red herring. We just don't know. So that's why we've got to find the timing. We need to know the timing before we can address the question of therapy.
And again, related to that question, indeed, I also believe PCDH19 might be involved in activity-dependent processes. So the refinement during adolescence, synaptic refinement during adolescence is basically activity or experience dependent. And indeed, PCDH19 gene, expression of the gene changes depending on the activity. So that's why activity-dependent processes may also involve PCDH19, including synaptic plasticity, and that's something we are also actively working.
I was just going to ask about the ASO approach. I think the most human data we have is the Dravet syndrome ASO trial. It's looking at this splice site. Is it safe to, I won't say assume, I'll say assume, that this knockdown approach, right? Is this a potential theoretical advantage? It's easier to knock down something, right? So we can end up some optimism here, right?
Yeah.
And I mean, in Dravet syndrome, we're trying to look at this, right? The splice site transcript, and we're seeing the data looks good, but it's not, you know, it's not everyone's responding to these therapies, and we're talking about seizure reduction, not seizure cessation. So I'm wondering if maybe we can be, you know, the optimism of the knockdown approach because of the unique pathogenesis.
I agree 100%, Joe. A knockdown or knockout approach is so much easier than trying to restore a gene's function. Make a broken gene and fix it again, that's tough. And that would have to apply to many different mutations, and would be a very difficult exercise. But fortunately, predicate adherent 19, we can use the opposite approach, which is to break the good copy of the gene that remains, and that we know from a lot of evidence that that could restore function. So that's an advantage of predicate adherent 19, sure. Yeah. And actually, one more thing, I'll just quickly say that the age of the CRISPR therapy has now started. So late 2013, in 2 countries have now approved for the first time CRISPR therapy for sickle cell disease, so a different disease entirely. But that's a process that started in 2012 when CRISPR was first discovered as molecular scissors, and now it's 10 years later, it's in the clinic, which is pretty amazing. So there's lots of good stuff going on in this space.
All right, on that note,